Welcome back to yet another of my series. We are going to be looking at bioremediation. First of all, let's look at what remediation is. And we say that remediation is the process of reducing or removing the toxic effect of waste to an initial state or to a level below concentration limit established by regulatory authorities. So whenever you're talking about remediation, it has to do with removal of toxic effects, either by removing the toxicant itself or by converting it into states that it will no longer be harmful to the body. Now, methods of remediation. There are two methods used in remediation: the physical method and the biological method. The physical method, which is the use of physical means to remove pollutants or waste in an environment, and this includes burning, addition of chemicals such as surfactants and such as detergents, and even removing them to a new location. Okay, now these are physical methods. What are the advantages of this physical method? The major advantages include they have broad spectrum of applicability. You can apply physical method in a very wide range of pollutants. They are high efficiency. They are high specificity of application for individual pollutants. Okay. What are the disadvantages of this physical method? They are usually very costly. They are expensive methods. They also produce large amount of waste, including hazardous waste. We have the problem of process control, especially in case of in situ techniques. Okay. The biological method is the use of biological means such as living materials or products of living materials such as biosurfactants, enzymes, and so on to remove pollutants or waste in the environment. And this method includes the bioremediation, the phytoremediation, the rhizoremediation. Okay, any method that you will use a living product or a living organism or a product of a living organism to remove waste is a biological method of remediation. Now, recent studies in molecular biology and ecological offers opportunity for more efficient biological process to clean up polluted environments, especially water and land. Now, bioremediation allows natural process to clean up harmful chemicals in the environment. Microscopic bugs or microbes that live in the soil or groundwater like to eat these harmful chemicals. Okay, when the microbes completely digest these chemicals, they change them into water and harmless gases such as carbon dioxide. Now let's define bioremediation. Bioremediation is a branch of biotechnology that employs the use of living organisms like microbes, bacteria to remove the contaminants or pollutants or toxins from water, soil, and other environments. Bioremediation is used to clean up oil spills or contaminated groundwater. Or we can say that bioremediation is a process used to treat contaminated media, including water, soil, and subsurface material, by altering environmental conditions to stimulate growth of microorganisms and degrade the target pollutants. Okay, so you have seen from definition, we are just bioremediation has to do with using living organisms, whether the organism itself or the products of the organisms, like I've said before. It is a biological means. Okay, now what we are trying to explain is that the microbe in the environment digests or feeds on the pollutants, say oil. Okay, you will see the microorganism engulfing the oil, now converting it to water and harmless gases, and then released to the environment. Okay, so we say bioremediation is an option that offers the possibility of destroying and rendering harmless various pollutants using natural biological activity. Microorganisms may be indigenous to a contaminated area, or they might be isolated from somewhere and brought into the contaminated site. Contaminant compounds are transformed by this living organism through reaction that takes place as part of their metabolic process. Biodegradation of compounds is often as a result of actions of multiple organisms. Microorganisms must enzymatically attack the pollutants. Then bioremediation can be effective only where the environmental conditions permit microbial growth and activity. Manipulation of these environmental parameters needed for microbial growth and degradation to proceed faster. Okay, so this sense is what the organisms in the environment do during bioremediation. Now, what are the factors that can affect bioremediation? The control and optimization of bioremediation process is a complex system of many factors. One of the factors that can affect it is the existence of a microbial population in that environment. Availability of contaminants to the microbial population, then the environmental factors such as soil type, temperature, even salinity, pH, oxygen availability, or other electron acceptors, and above all, nutrients. Okay, for the organism, because the organism normally uses the pollutant as carbon sources, so it will still need the availability of nutrients such as nitrogen, phosphorus, and in some cases sulfur, because most of um, these nutrients are used for amino acids and nucleic acids production in the organism, so they must be available for remediation to be effective. Now let's look at them. The microbial population. Microorganisms can be isolated from almost every environment. The microbes will be adapted and can be adapted and grow at sub-zero temperatures, as well as extreme heat, desert conditions, in water and in excess of oxygen, and the organism can grow in the absence of oxygen, okay, and they can grow in the presence of hazardous waste, okay. The main requirements are energy sources and the carbon source. Because of the adaptability of microorganisms and other biological systems, this can be used to degrade or remediate environmental hazards. Now, types of microorganisms. We have the aerobic organisms, the one that grow in the presence of oxygen. They have high demands of oxygen. We have the anaerobic, the one that grow in the absence of oxygen, and thereby they can even use other electron acceptors, something like carbon monoxide, something like hydrogen sulfide, and so on and so forth. Then we have the methelotrophs. These ones utilizes methane as a carbon source. Okay, now they can use to clear a very wide range of compounds, which include the chlorinated aliphatics, trichloroethane, and 1,2-dichloromethane. So these organisms can take care of such. Now, the different methods in bioremediation. Now, in this method, we have the biostimulation, we have the bioaugmentation, we have implicit bioremediation, and we have enzyme technology. These are the methods that we use. Now, let's look at biostimulation. Although microorganisms are present in contaminated soil or environment, they cannot necessarily be there in the numbers required for bioremediation of the site. Their growth and activity must be stimulated. So in biostimulation usually involve the addition of nutrients and oxygen to help the indigenous microorganisms. So in biostimulation, we add nutrients, maybe in form of nitrogen, in form of phosphorus to break down um, the to break down the pollutants. These nutrients are basically building blocks of life and allows microbes to create the necessary enzyme to break down the contaminants. All of them we need nitrogen, all of them we need phosphorus and carbon, and to some extent sulfur. Okay, now carbon is the most basic element for life form and is needed in greater quantities than other elements. In addition to hydrogen, oxygen, nitrogen, it constitutes about 95% of the weight of the cell. For degradation, it is necessary that the bacteria and the contaminants come in contact. This is not easily achieved as neither the microbe or the contaminant are uniformly spread in the soil. So most of the times we begin to see localization of either the microbes or the contaminants. In some areas where there are some more carbon source, we see more organisms and so on. They are not evenly distributed. Now, some bacteria are mobile and exhibit a chemotactic response, sensing the contaminant and moving towards it. Other microorganisms such as fungi grow in filamentous form towards the contaminant. So there must be a way that you will use, to bring them in contact. Otherwise, bioremediation will not take place. It is possible to enhance mobilization of contaminant utilization, of mobilization of the contaminants utilizing some surfactants such as SDS, that's sodium diacetyl sulfate. When you apply some contaminants, especially in oil polluted environment, it can help solubilize the oil and make it available to the organisms. Now let's look at bioaugmentation. Bioaugmentation is a practice of adding active growing specialized organism strain into a microbial community in an effort to enhance the ability of the microbial community to respond to the process fluctuation or to degrade certain compounds, resulting to improved treatment. By changing microbial community by changing the microbial community to include specific microbes, the characteristics of the community can be improved. In a nutshell, in bioaugmentation, we apply organisms. In biostimulation, you apply nutrients. Okay. Now, for instance, if we have an environment that is contaminated with something like crude oil, I will be mentioning crude oil more often. And then in the environment lacks nitrogen. One might naturally want to introduce nitrogen fixing bacteria, okay, or phosphorus solubilizing bacteria into that environment so that they will now produce to help or even introduce some other organisms that can break down the crude oil. Now, the idea is to help the organisms in the environment to do the work. Okay, in stimulation, you are causing the organisms to grow and do the work. In stimulation, in augmentation, you are helping producing organisms. So, major difference in stimulation, nutrients are added. In augmentation, bacterial cells are added. The purpose of bioaugmentation is to supply the existing microbial community in order to improve its functionality. Bioaugmentation offers many advantages over traditional technology platforms like chemical equipment and other consumables that have been used in secondary waste water treatment system for decades. Okay, now intrusive remediation or natural um, remediation, or what we call natural attenuation. Okay, natural attenuation is known as intrusive remediation or natural remediation. Is a passive remedial approach that depends on natural processes to degrade and dissipate contamination in soil or in groundwater. In other words, in intrusive remediation, you are allowing the indigenous organisms to actually do this work. Now, natural attenuation process include physical, chemical, and biological transformation. Example: aerobic, anaerobic bioremediation, co-metabolism, dispersion, volatilization, oxidation, reduction, and absorption. Okay, this can happen naturally in the natural attenuation. Aerobic and anaerobic bioremediation in bioremediation, that's intrusive remediation, is believed to be the major process that accounts for both contamination, both contamin containment of the dissolved contaminant plume and reduction of the contaminant concentration. Okay, now using both aerobic and anaerobic bioremediation, that is the organism, they are here to contain the organism and contain the contaminant and reduce its concentration. Aerobic bioremediation relies on dissolved oxygen as electron acceptor by the use of subsurface organisms, while anaerobic processes revert to a variety of bioremediation mechanisms that uses nitrate, ferric ion, sulfate, and carbon dioxide as terminal electron acceptors. Anaerobic bioremediation dominates the interior of a contaminated plume. In other words, inside that plume where oxygen is not available, the anaerobic processes takes place. Now, environmental conditions and microbial competition will ultimately determine which anaerobic bioremediation process will dominate. But in typical aquifer, it is devoid of dissolved oxygen. Denitrification typically occurs, followed by ion reduction, sulfate reduction, and methanogenesis. Okay, so in the environment, especially when there is no dissolved oxygen, you will begin to see processes like denitrification taking place. Okay, ion reduction, phosphate reduction, and then the production of methane. Now, advantages of intrusive bioremediation over other engineered remediation technologies include: one, contaminants are ultimately transformed into initials byproducts, example carbon dioxide and water, most of the times not just transfer from one phase or location within the environment. So number one is that they break down the pollutants to a point where they are not destructive. Now, intrusive bioremediation is non-intrusive and allows continuous use of infrastructure during remediation process. It is less costly and it is not subject to limitation imposed by mechanization of remediation equipment. Now, because you are allowing the organism in the environment to do their work, it does not really matter. Do you understand? It doesn't depend that you must get a mechanized equipment. Okay. Now, to support remediation by intrusive bioremediation, the proponent the proponent most scientifically demonstrated that degradation of sites contaminated is occurring at a rate sufficient to be protective of human health and the environment. There are three lines of evidence that can be used to support intrusive remediation of chlorinated aliphatic hydrocarbon. These include: 1. You observe reduction of contamination concentration along the groundwater path from the contaminated oil location. Document loss of contamination mass at the field scale, and then the microbial laboratory data that supports the occurrence of bioremediation or biodegradation. Now let's look at enzyme in bioremediation. That's enzyme bioremediation. First of all, let's actually consider what happens in bioremediation of wastes. You have the waste. The waste is being attacked by fungi, bacteria, even plants to produce green earth and to reduce it. Okay. Now, we are not really going to be defining an enzyme, but I believe we all know what enzyme were. Enzymatic bioremediation, isolation of enzyme may also be used to transform contamination to less toxic or non-toxic compounds. Now, normally what is used is extracellular enzyme. These are enzymes that are being secreted outside the body of the organism, such organisms as rot fungi and others during fermentation process. And also the one produced by plants during phytoremediation. And in situ, the use of plants and their enzyme system, their roots and associated organisms to degrade harmless pollutants present in different environmental systems such as water, soil, sediment, groundwater, air, and so on and so forth. Now, little background that enzymes are organic catalysts that facilitate the bioconversion of substrates to products by providing favorable conditions that lowers activation energy. This one, enzymology. The region of the enzyme that directly involved in catalysis is known as active site, and we are trying to look at it. And the active site, the enzyme, okay, the substrate binds to the active site, and then a lot of reaction goes on, and transformation and products are formed. Now, extracellular enzyme in bioremediation. Extracellular enzyme, like I said, refer to those enzymes that are either secreted by microbes such as white rot or those that enter the aqueous phase during aerobic submerged fermentation. Such enzymes are naturally produced by the microorganism or the microbe, and then they are harvested. Enzymes from the white rot have been shown to be effective degraders of phenol, PCBs, PAHs, and dyes. Okay. Advantages and disadvantages of extracellular enzyme. 1, they can work in multiple environments. They can be recovered and recycled. They can be used with different substrates. They can be biodegradable. 2, it is difficult to maintain enzyme in the concentration because the enzymes are soluble. It is difficult to optimize. Limitation over success. It has a lot of limitations, and the amount invested in trying to produce and isolate these enzymes are very expensive. Soluble and immobilized enzyme. Now, mobile enzyme can be added at a single point and they spread due to diffusion and dispersion. They flow to underground surfaces. Now, we have immobilized enzyme, which we looked at when we are looking at enzyme immobilization. Okay, the enzymes can be immobilized into a carrier, which can be granular, fibrous, or in a tube, or even in a membrane. In general, immobilization makes the enzyme more resistant to temperature, pH, and substrate concentration swing, giving it a longer life and higher productivity per activity unit. Plants and their associated enzyme remediation. Okay. Now, the involvement of plants in bioremediation of pollutants is often called phytoremediation. The process of phytoremediation is an emerging green technology that facilitates the removal and the degradation of toxic chemicals in soil, sediments, groundwater, surface water, and even air. So the use of plants to remove pollutants can be known as phytoremediation. Now let's take a look at some things that happen in plants in phytoremediation, considering the rhizosphere. Okay, now there you can see respiration. The left side of the talk about respiration, where CO2 and oxygen are being exchanged. Okay, during the breakdown of the contaminants. Also, there is also exchange of hydrogen ion and hydroxyl ion, because sometimes the pH of the environment is affected. Now these organisms produces oxidases and hydrolysis that degrade organic compounds. They also produce cytochrome P450s. The hydrolylation of aromatic compounds requires these particular enzymes. Okay, now the microbial enzymes which affects the growth and the physiology of an organism is also present in mostly the roots. Okay, and these organisms can be chelators. They help in the delivery of plant nutrients. They can be bacteria acid deaminase, regulate ethylene levels in plants. We have these organisms as producing nitroreductases that reduces nitro compounds that is found in aromatic compounds. Okay, we have also the phosphatases that cleaves phosphate groups from organophosphates and even from soil. You have the nitrolylase that cleaves cyanides from aromatic and aliphatic nitrites, and then the lactase that degrades various aromatic compounds. All these could be found in the roots of the plant that is used for bioremediation processes. Now, let's look at some of these enzymes that are used in bioremediation. Now, enzymes from white rot fungi have been found to be very capable of degrading large amounts of different contaminants. White rot fungi are unique among eukaryotes because they are able to cleave the carbon-to-carbon bonds in contaminants such as polyaromatic hydrocarbons. During the secondary metabolite of plant life, white rot produces and secretes lignin peroxidase, magnesium peroxidase, and lactase. Each of these enzymes can catalyze the 1-electron oxidation of phenol and non-phenolic substrates. I hope you take note of the enzymes. The result of this production of cation radical intermediate, which can be used to further oxidize non-phenolic substrates. The lignin peroxidase are homoproteins which catalyzes reaction in the presence of hydrogen peroxide. In other words, hydrogen peroxide acts as a catalyst to this enzyme. Okay, but it's not a catalyst per se. Now, the LIP is a very effective in bioremediation of PAH, such as benzoapyrene. We also have the magnesium peroxidase, and we say that this is also a hydrogen peroxide dependent enzyme, but it can only oxidize organic compounds in the presence of manganese 2 ion. Now, in that process, it will oxidize the manganese 2 ion to manganese 3 ion, which acts as an obligatory oxidation intermediate for the oxidation of various compounds. The manganese 3 ion migrates away from the enzyme and starts the oxidation of lignin and other compounds. Okay, now let's look at what happens at the root of this fungi. A hyphae of the fungi, you begin to see that sugar, acetyl, hemicellulose, which is part of wood, is being transferred. And what will this organism do? It's going to produce lacase and other peroxidases, okay? And these peroxidases will break down lignin and break down lignin and make them soluble, okay? Now, it can also produce some other co-oxidants, unsaturated fatty acid and thiols, okay? Now, because it has this enzyme manganese peroxidase, which is dependent on hydrogen peroxide, is going to oxidize magnesium 2 to manganese 3, and this manganese 3 is going to be transported to where you have the pollutant, okay, which is going to help to oxidize some of the pollutants that we have. Okay, now lacase, it says it's a multi-copper oxidase that catalyzes one electron oxidation of substituted phenol, aniline, and aromatic thiols to the corresponding radicals with a concomitant reduction of molecular oxygen to water. Now, the radicals produce polymeric products by self-coupling or cross-coupling with other molecules. And how does it do that? It does that by dechlorination or demethoxylation or decarboxylation processes, which occurs during the coupling process. The polymerization of different substituted substrates may also occur. This enzyme appears suitable for versatile catalysis, very useful for the application of several biotechnological processes. Now, another enzyme that we can use in bioremediation is the hair root peroxidase. The hair root peroxidase is a peroxidase which is secreted by the hair root of horseradish plants and can catalyze the oxidation of compounds such as phenol, biophenol, aniline, benzidine over a large range of pH and temperature. Extracellular enzymes such as proteases, amylase, and lipases are produced during the aerobic fermentation of organic matters by yeast or other microbes. Environments such as wastewater requires a different types of enzyme, and this enzyme, rather than catalyzing the oxidation of recalcitrants, catalyzes the degradation of organic matter. And that is some of the problems that we face in bioremediation, and that process is the focusing of your enzyme or your microorganism. When your microorganism is meant to break down crude oil and you introduce it into the crude oil, and this microorganism now finds other carbon sources that are suitable, it will leave the crude oil and begin to consume the alternative sources. Bioremediation of textile effluents using enzymes can act on specific recalcitrant pollutants to remove them by precipitating and transforming to other products. White rod fungi are able to degrade dyes using the lignin peroxidase and magnesium dependent peroxidase, okay? And that is why these enzymes are used in textile industries. The magnesium peroxidases can dechlorize azo and anthraquinone dyes. The lignin peroxidase can dechlorize methylene blue and azo B dyes. Then the versatile peroxidase has been recently described as a new family of lignolytic peroxidases together with lignin peroxidase and manganese peroxidate isolated from microorganisms. Now, recalcitrant dyes could be successfully decolorized by peroxidases in the presence of some suitable redox mediators. For you to remove dyes, it can be done in the presence of some peroxidases. Okay, now these peroxidases can be used in textile industries in the removal of dyes or stains. Treatment of recalcitrant products by using enzyme redox mediator system will be significantly useful procedure for targeting number of dyes with diversified structures. Now, lacase can completely decolorize bromophenol blue and malachite green, both triphenylmethane dyes, and partially decolorize the color orangey and amaranth, both are azo dyes. Immobilization of this enzyme on alumina increases its stability and is made less effective by inhibitors such as halide and dye additive. Immobilized lacase was also able to decrease the toxicity of this dye by up to 80%. Enzymatic decoloration of industrial dye is a big challenge due to large diversity of chemical structure. Yes. Now, this challenge is as a result of the nature of enzymes. Enzymes are specific, and when you have a lot of chemicals with different structures, the enzyme might not be specific, except some that might even work using induced feed mechanism. Enzyme offers several advantages such as greater specificity, better standardization, easy handle and store, and no dependence on microbial growth rates. That's one major thing about enzymes. Once they are isolated, they do not depend again on whether you're growing the microorganism or not. Now, a major obstacle that we have to be overcome is the long contact required for decolorization to occur. Remember, enzymes are soluble. Now, enzymes present environmental advantages against chemical and microorganisms. We are looking at the advantages of these enzymes in the environment over using chemicals or microorganisms. When we say biotransformation does not generate toxic side products, as often the case of chemical and some microbial process. Now, why we do this is that most of the times these microorganisms, they might be virulent, they might have a way of causing illnesses, of which organism that does that should not be used for biotechnological approaches. The enzymes are digested in situ by indigenous organisms after treatment. The requirement of an enhanced bioavailability by introducing organic solvent or surfactant is much more feasible for an enzymatic point of view than the whole organism. Okay, the production of the enzyme at a high scale with enhanced stability and or activity and a lower cost of feasible by using recombinant DNA. In other words, what you are just trying to explain here is that when you use a recombinant DNA technology to get this enzyme, okay, that you can generate enzymes that has a wider scale of application other than using chemical agents or some microorganisms. However, a more extensive effort is required to overcome several bottlenecks. And that bottlenecks, like I said before, is the high cost of enzyme. To isolate enzyme is very expensive, low activity or stability under given conditions, low reaction yield, and the low biodiversity of organisms screened so far. Now let's now look at phytoremediation. Phytoremediation, we have defined it as the use of plants, and there are 5 methods involved in phytoremediation, which include phytostabilization, phytotransformation, phytovolatilization, phytoextraction, and phytostimulation. There are 5 of them, and I will want you to actually go ahead to look at each and every one of them. Okay, phytodegradation, where the plant helps to degrade pollutants. Phytovolatilization, where the plants help in converting them into um, volatile substances. Phytoextraction, where the plant helps to extract these pollutants by absorbing them into the plant. Phytostabilization, where the plant helps to convert the chemical into a more stable and unpolluted form. Now, phytostimulation, where the plant helps to produce substances that can stimulate the growth of plants. Now, phytoremediation, we say that is an effective and low cost and environmental friendly soil cleanup of heavy metals and organic compounds. That is where we use them most at times. Pollutants are absorbed in the roots, and thus plants are removed, disposed, are burnt. Okay, sunflower plants are used to remove something like cesium and strontium from the pond at nuclear plants. Okay, the idea is that you use the plant as they are growing, they are absorbing these heavy metals, and then what you do is you can now collect the plant and probably go to an incinerator, burn them, dissolve the ashes, and get back this metal in solution. So it is no longer there in the environment, but it can be in solution. Transgenic plant with exogenous methionine, a metal binding protein, is used to remove metals and heavy metals from the environment. Now, bioremediation of metal contaminated environment. So in metal, the mechanism is that it uses metal immobilization or metal solubilization, or what we call biobleaching. Now, you can use organic acids, seed rufous, and root exudates. Okay, can actually help to solubilize these metals. Now, under immobilization, there could be complexation and precipitation. Okay, in precipitation, you are converting the metals into inactive forms or into a non-reactive form so that they can precipitate, and that precipitation, you have the hydrogen sulfide producing bacteria can do that. Okay, there are also exopolysaccharides production that can also help in complexing these metals. Okay, now where are the sources of these heavy metals? We all know mining activity, atmospheric deposition, waste disposition, agrochemicals, industries. You can just mention so many of them. Now, biosurfactants. Now, biosurfactants are just surface active compounds, okay, that are used to reduce the surface tension of oil. So biosurfactants are chemical compounds characterized by the hydrophobic and hydrophilic, that is the non-polar and polar region in one molecule. In other words, we say that this molecule are amphipathic in nature. Okay, biosurfactants have been produced from bacteria, fungi, yeast, and they have been classified into glycolipids, lipolipids, phospholipids, glycoproteins, polymeric biosurfactants. A lot of biosurfactants have been produced over the years. Now, some organism has actually been genetically modified. Okay, so a genetic modified pseudomonas can produce biosurfactant. This new strain can produce the glycolipid emulsifiers. Okay, it can reduce the surface tension of oil. I have said that, and reduce the interfacial tension, promoting degradation of oil. Okay, in other words, in bioremediation, especially of oily surfaces, we make use of these biosurfactants. Now let's still look at these types of bioremediation. Now the other one, we look at the methods of bioremediation. Now let's look at the types. There are two types of bioremediation. We talk about the in situ bioremediation, we talk about ex situ. As the name implies, the on-site remediation and the um, off-site remediation. Now on the on-site, we've talked about the intrinsic bioremediation and the engineered bioremediation. And the engineered bioremediation, we talk about biosparging, bioventing, biosporing, biosloping, biostimulation, bioaugmentation, natural attenuation. We've looked at that. Now, ex situ, where you remove the contaminant from the site and then take it to the place that you're going to do the whole remediation process. Now you can look at slurry phase bioremediation and solid phase bioremediation. In slurry phase, you make use of a bioreactor. Okay, the slurry phase can also be known as a liquid phase bioreaction. Are we following? Yeah. Now, the solid phase, you can talk about the biopiling, land farming, composting, biofilter, and so on and so forth. Now, still on this type, in situ remediation, the cleanup is done by direct contact of the organism and the contaminant. But there are advantages and disadvantages of in situ. In situ is low cost, minimal site disruption. You don't disrupt a lot of sites. Simultaneous treatment of contaminated water and soil, you can do that simultaneously. Then minimal exposure of public and site personnel. But the disadvantages is it's time consuming. Bioremediation takes a lot of time when you compare it with even a physical method. It's seasonal variation because you're dealing with organism and this organism responds to seasons. The season at which you're doing the remediation is very, very important because when you do in dry season where the water availability is quite low, it will halt remediation in certain places. Okay, now you have probabilistic addition of additives. It causes some problem in situ remediation. Now the in situ, there are two types in situ remediation. We've talked about that. In in situ remediation, there are two types: intrinsic and natural attenuation and enhanced bioremediation. Okay. Now, in the enhanced bioremediation, we talked about biosimulation and bioaugmentation, and these methods and the stimulation, you can do bioventing, where you stimulate by actually adding air. You can look at land farming, okay. You can look at phytoremediation and air sparging. All these are methods of biostimulation. Now, intrusive technologies, they have factors that can affect this. The first one is moisture in the soil, temperature, pH, nutrient availability, presence of electron acceptors, okay, something like nitrate and sulfate electron acceptors. Then the concentration of the toxins. When the amount of organism does not match the concentration of toxin, there could be a problem. It might not necessarily work. It might overwhelm the organism or even to greater extent kill the organisms. Engineered bioremediation, it can be spoiled on via addition of fertilizer or by manipulating other components to increase the bioavailability within the medium that is in biostimulation. Or you can do it by, adding other organisms to help. That's your engineering other organisms there. Now, ex situ bioremediation, we have talked about it. We say is ex situ conservation, literally meaning off-site conservation, the removal. You collect the waste and take it to a facility, which can be classified into the solid phase and the slurry phase treatment. Now, solid phase treatment include organic waste, the kinds of things you can use in solid waste, organic waste, domestic waste, industrial waste, and agro waste. We use the traditional method of composting. And what do? You get this waste, put it in the ground, and bury it. That is composting. Okay. Composting is self-heating, substrate dense, manage microbial system stability for the treatment of large amount of contaminated waste. Hazardous compounds reported to be removed from composting are aromatic hydrocarbons, halogenated compounds. Okay, what actually can affect composting? The same thing. Moisture, if your compost is dry, degradation will not take place. Okay, so moisture content is very important, pH is very important, temperature is very important, soil nutrients is very important, even soil particle size are very important, and also the concentration. Now, composting can be done either open land system or in the closed system. The open land system is an inexpensive but effective way to manage, but the closed treatment is expensive but manageable. In open land, you might not manage, you cannot control it, but in closed system, you can control it. And what you mean by the open land is what you have seen. Okay, you can just do it, allow degradation to go on in the open land, which means you can't control anything. But in a closed system, you can control, you can till the ground, turn the process so that the organism can go in contact with the contaminant. Now, slurry phase system or liquid phase. Okay, the contaminated material and organisms and water are formulated into a slurry within a bioreactor or a fermenter. Do you understand? So you have a fermenter, you put the organism, you put water, and then you make a slurry so that there will be continuous, touch. Now, types of slurry phase, you have the rated lagoon. Okay, you have the closed system. In the bioreactor, the pH and the temperature, nutrients, everything is mixed. It's a closed system. Okay, and sometimes we even have to mill the pollutants to reduce the size. Okay, so that the organisms can easily degrade them. And sometimes you can add surfactants, so that some of the oily ones can be dissolved and have a better contact with the organism. Now, when you look at these factors, the key point of the solid phase factors affecting it, they are the same. The moisture content. It's not that there's a way you increase the water content, okay, it will affect bioremediation or biodegradation. Now, the pH, the temperature, the soil nutrient, the reactor operation, toxin concentration, microbial population, and mixing. How often do you mix? Okay, all these things are factors that can affect. Now, solid phase bioremediation, this time around, you're not adding any water. So we're saying solid bioremediation treatment can be conducted in lined land treatment unit or in composite piles. In line treatment unit consists of a prepared bed reactor with a chilled collecting system and irrigation and nutrient delivery system. Okay, now in this place, is the whole thing is an open environment, but you're going to introduce an irrigation here so that you are taking care of the moisture content. You have to have a way of delivering the nutrients to the organisms. Now, this unit may also contain an air emission control equipment. Now, the soil is placed on land lined with and impervious layers such as soil, clay, or synthetic liners. You also have bioventing. Okay, yeah, bioventing. Um, we say we use relatively low flow erosion techniques to enhance bio degradation of soil. You can actually download some pictures of bioventing. Okay, in other words, in bioventing, all you do is you dwell so much on supplying air into the contaminated soil so that aerobic organisms can help to do the work. We have land farming where you till the area, that when you are tilling the area, it helps the soil to come in contact with the organism, and the organism is also coming in contact with the pollutants. So in tilling, it helps. Land farming, it helps. Okay, we say it's an S2 process which include land farming, which involves spreading contaminated soil in a large over a large area. Bioremediation may also be conducted in a bioreactor in which the contaminated soil also this is not supposed to be here, but just look at the now biopile treatment is also part of it, and then the use of bioreactors. Okay, we've talked about composting. Now we can compare both in situ and ex situ reaction. Wow, this lecture is really a long one. In situ, like you said, it means in the original place, ex situ outside. In situ methods are carried on site, while ex situ methods are taken to the laboratory. In situ methods are applied to a large population. Then the ex situ method is applicable to a smaller population. Experimental conditions are difficult to maintain in situ, but in ex situ you can maintain the experimental condition. In situ is less expensive, ex situ is very expensive. In situ do not require much equipment and it is less laborious, while ex situ requires a lot of equipment which can be labor intensive. The in situ requires a large area while ex situ requires a smaller area, I think. So, to summarize the strategy, we've looked at some of them. You can see the technology, whether in situ, ex situ, and our bioreactor. For example, in situ bioremediation, we have the biospaging, the bioventing, bioaugmentation. Okay, ex situ you have land farming, composting pile, and then bioreactor you have slurry reactor or aqueous reactors. So we have their benefits which you need to go through. Benefits of in situ, we have talked about it. Most cost effective, non-invasive, relatively passive, natural attenuation process treating soil and water. You can do that together. Limitation, environmental constraint. You can control the environmental um, properties. Extended treatment time and monitoring is difficult. Factors to be considered when you're using in situ: you will consider the biodegradability of indigenous organisms. You will consider the presence of metals because some metals might harm the organism and other inorganics. Environmental parameters, you must consider them: biodegradability of pollutants, chemical solubility, geological factors, and distribution of the pollutants. Like I said, distribution of the pollutants is quite necessary. Okay, ex situ, you have the same factors, but the limitation of ex-situ is that space requirement, extended treatment time, needed control, abiotic losses, mass transfer, and so on and so forth. Advantages of bioremediation. Bioremediation is perceived by public and as an acceptable waste treatment process. Microbes able to degrade contaminated contaminants increase in number when the contaminant is present. When the contaminant degrades, the biodegradative population declines. So you don't have anything like overpopulation, because once they take care of the system, they naturally die off. It is safe as the residues of the treatment are harmless, which include carbon dioxide, water, and cell biomass. It is useful for complete destruction of a wide range of contaminants. They eliminate the chance of future liability associated with treatment and disposal of contaminated materials. Now, instead of transferring contaminants from one environmental medium to the other, for instance, from land to water or air, they completely destroy the target, the pollutant is possible. What I mean, for instance, when you're using the physical method of burning, all you do is that you convert, the pollutant from being on the ground to being in the air, because more other pollutants are being formed. Uh, it can it can it can often be carried on site without disrupting the normal activity. No need to transport waste off site. Okay. It does not require too much sophisticated equipment. Bioremediation can prove less expensive than other technologies that are used in cleaning up of hazardous waste. Disadvantages. Bioremediation is limited to compounds that are degradable. Not all compounds are susceptible to rapid and complete degradation. There are some compounds that are recalcitrant. They are not easily degraded. Now, biological processes are often highly specific. Important side factors required for success include the presence of metabolically capable microbial populations, suitable environmental growth conditions, and appropriate level of nutrients and contaminants. So these are if you don't have enough organism, the degradation will not take place. If the contaminations overwhelms the organism, degradation will not take place. Okay. And then you have to maintain some environmental conditions just like that. Those things are disadvantages of bioremediation. Now, it is difficult to extrapolate from the bench and pilot scale studies to full scale operation. Research is needed to develop and engineer bioremediation technologies for complex mixtures of contaminants that are not even dispersed in the environment. Bioremediation often takes longer than treatment options such as excavation, removal of soil, and incineration. It takes longer time. What are the limitations of bioremediation? The problem of on-site remediation by microbes are often failed for two major reasons. First, the introduction of microbes cannot establish a niche in the environment. In fact, these introduced organisms often do not survive in the new environment beyond a few weeks because they don't have suitable carbon sources. Secondly, there are difficulties in delivering the introduced organism to the site of contamination, because organisms like contaminants can be strongly absorbed by solid surfaces. Okay, the overall scenario in current status of bioremediation is given in a table below. I'm sorry, the table is not there, but we have mentioned the basic things. Now, the adverse effect of crude oil spill. One is that they cause spreading, and when we talk about spreading, it causes the oil to spread over a large area of both land and water, even when the water is not flowing. Okay, oil has a way of spreading, and why does it have the way of spreading? Because of the gravitational force and then the surface tension, and even also the temperature of the water can encourage spreading. Another factor is evaporation. Okay, as evaporation occurs, some of the volatile oil, escapes, leaving the ones that might be, recalcitrant. Okay, and then the type of oil, affects this evaporation rate. Now, another thing is that emulsification can help. Okay, because it will help to solubilize, the oil in water. Now there is petrophoto oxidation. Now when there is a spill, the oil flows and coming in contact between oxygen and water can lead to photo oxidation. Okay, so when that happens, it can transform the oil into some other solutions. There is also bacterial degradation. Okay, and when this happens, we say that petroleum contains a whole lot of compounds: carbon, hydrogen, sulfur, oxygen, and even nitrogen compounds. Okay, now these microorganisms can begin to break this oil, that even when you try to recover, you will find out that the oil is no longer what it used to be. Now, what are the biological effects of oil leaks in um, aquatic environment? We say there are effects, and these effects, is as a result of some combination of factors. And what are these factors? One is the quality of oil and the quantity of the pollution, the duration experienced by the organism. Is it just the fresh one? Because most of the times when oil comes in contact with organism, it's like this: the oil is making the organism to exist in a new environment, so it need to adapt to that environment. So the duration is very, very important. The state of oil, whether the oil is just produced or weathered or refined product, is necessary. Now the season is also necessary. What season is it? Is it the season where these organisms are multiplying on their own, or is it a season of dry, or where the environment is dry, and so on and so forth? Now the habitat and the natural stress to the organism is subjected. Like I said, once the oil comes, it makes that place feel like a new environment, and then the organisms need to adapt. We say the manner of the effect of these factors as listed below. Now, due to all these effects that we have mentioned, we will notice that there could be direct liver toxicity. That is, the organism begins to die. Or two, sublethal destruction of physiologic and behavioral activities. Now, because this is a new product, the organism begin to adapt, begin to try to begin to feed on new things, and so on and so forth. And like we said, because of these changes, there could be death of the organism also, because there is interference in their feeding and also interference in their reproduction. because the presence of some of these pollutants might halt reproduction. Now, there is direct coating and painting. Sometimes, you see this polluted oil spill environment. The oil is so much that some of the birds, that and dogs, you see them where they cannot even fly again because the oil have coated them. Now, another thing is that it causes the entrance of hydrocarbons into the food chain, and this is not very okay because it can lead to, if the hydrocarbon is cancerous, it means you have introduced a cancer causing agent into the food chain. Now, some of them will begin to migrate to different places. The fishes can begin to swim far places. Then the eggs and larvae that do not have the capacity to float to swim might die off and reduce population. Wow, this one seems to be a long lecture, but if you have any question, please send it to the chat group and we can answer it from there. Thank you very much.